So a natural extension of what we've been doing so far is to take the sequence analysis that we've been doing and apply it in a larger context, right? So you have a DNA sequence, and now you have a DNA sequence that really becomes a chromosomal sequence, and you put all the chromosomes related to a species and you get a genome. So what we're going to do is extend everything we've learned related to sequences and begin to apply it to genomes. So we'll be looking at some fundamental conceptual ideas, and also we'll also be looking at some resources that are available that are actually quite invaluable. So we're looking at here the bioinformatics of the genome. And one of the first aspects of the bioinformatics of a genome is, and this particular class will go into maybe several modules where we're going to start with the basic notions of how do you determine an unknown sequence. Okay, this whole notion of what is sequencing, to be able to say I can now extend that and I should be able to determine the sequence up to a genome, okay, of a genome. And that is really the state of the art right now. It is this notion of next generation sequencing. So what is sequencing? Sequencing is a method to determine the exact order of nucleotides or amino acids in a DNA or protein sequence, respectively. Okay, so this is the exact sequence. You have an unknown sequence. You want to determine its primary structure. That is sequencing. So here we have a sequence of a nucleotide. It's a nucleotide. It's a DNA sequence. It's a fragment of a DNA sequence. And let's say, for example, the above sequence comes from a particular species, right? Right now, it is just a sample sitting in an Eppendorf tube. How do you determine what constitutes this sequence? What is the order of the adenine, the thymine, the guanine, and the cytosine? And the procedures required in order to determine this particular order of its primary structure is called sequencing. So let's look at the sequence. So the process of the sequencing is to take a sample sequence that is unknown and break it down into its constituent, just potentially a random fragmentation, and then find the sequence of each of these fragments, and we look at the two methods that we will use, and then reconstitute them to be able to say, okay, so we will talk about two different methods that have been in use, and one is probably fallen out of favor, and the second one, which is popular for the most time, is also, in my opinion, falling slightly out of favor as high throughput methodologies are beginning to be employed through next generation sequencing. But next generation sequencing is quite expensive, and everybody doesn't have those resources. So most labs use what is called the Sanger sequencing. But before we get to Sanger sequencing, let's begin to look at the Maxam-Gilbert. We call it P32 because the atomic mass number of stable phosphorus is 31. And when we talk about P32, that means it's radioactive, okay? And therefore, that's what we call it. That's what, when we talk about radioactive labeling, the phosphorus is labeled P32 on the 5' end, and available phosphorus. In one reaction vessel, you take formic acid, and you see the chemical formula, HCOOH, and it's used to break the purines at the 5' side of A and G. Okay, so there are different reactions that are occurring, and different reactions in each of those 4 vessels. Each of these reactions is used to break up in very systematic ways. And so you're going to get a lot of fragments, but because you understand exactly how you're breaking them up, the reassembly of these fragments into the sequence that you want to find out becomes easy. So, okay, just repeating that, in the first reaction vessel, formic acid is used to depurinate the 5' sides of the purines, which is adenine and guanine. In the second reaction vessel, methylated guanine's dimethyl sulfate, and we look at the reactions in a little more detail. Okay, so guanine. So now what happens is you break the A's, you fragment on the 5' end of A and G, but you can then further differentiate between the A and methylating G by using dimethyl sulfate, the guanines. In the third reaction vessels, the pyrimidines C and T are hydrolyzed using hydrazine. Okay, and we'll see what that reaction is, and we'll see exactly what hydrazine looks like. And in the fourth reaction vessel, the addition of sodium chloride prevents the thymine from reacting. Okay, so 4 different reaction vessels, 4 different reactions going on for the exact same sample fragment that is systematically fragmenting this sequence and the DNA sample, and we know exactly what, with great specificity, exactly where the fragmentation occurs. So once we understand how those fragments are, what those fragments look like, we can then go in and reassemble them into the sequence of interest. Okay, so let's begin to look at the chemistry. So formic acid is used to depurinate, right? So this is our purine, this is a ribose sugar, and this is the purine. And when you depurinate something using acidification, using formic acid, what we are doing is we are separating the purine group. We are removing the purine group, right? The adenine or the guanine, and the purine group, you know, it's a very specific reaction, and the break occurs here. So you're left with the purine, depurination, that is removal of the purine. The second reaction, methylation is guanine. Okay, and so here's what you see. This is dimethyl sulfate. Okay, sulfate group SO4, and dimethyl, 2 methyl groups CH3 and CH3. And here you have an adenine and a guanine. Now, again, a very specific demethylation occurs where in guanine, this group here, this nitrogen atom, is very specifically methylated. And that's why you know that a guanine is methylated completely differently from an adenine. Okay, it's this nitrogen atom on the 5-membered ring that is very specifically methylated in guanine, primarily because of the oxygen atom here. So there's some specificity associated with that, and that nitrogen atom is not methylated using dimethyl sulfate. Okay, that's the second reaction. In the third reaction, we have hydrazine. So you know H2N and H2. It's a very symmetrical compound. Hydrazine is used to hydrolyze and to remove the pyrimidine groups. Okay, remember the pyrimidine groups from our previous discussion. The pyrimidine groups are a 6-membered ring. And so this is, for example, the thymine residue, and this is the pyrimidine group. And what you're doing using hydrolysis, using hydrazine, is your pyrimidine group. So you're removing the pyrimidine group. And so what happens is when you break this here, you're going to get another, okay, fragment. And each of these fragments is going to have a very specific mass. And we can use electrophoresis, which is based on chromatography, to separate these fragments. And because we know exactly what the masses, or where you would expect each mass to show up, you know exactly what kind of masses is how the fragmentation occurs. So let's recap a little. Let's say, for example, you've got a sample DNA, and that's the fragment, right? ATTGAC, okay? And so what happens is that's the fragment that we have. That's the fragment that we're required to sequence, whose primary structure we want to determine. The first thing is we add a p32 at the 5' end. So now this is radioactively labeled. Okay, now you want to cleave at at specific sites. Now we've got four different reaction vessels. So one of this cleaves at G. Okay, one of these cleaves at G. So you'll see this is the full fragment, and so you're going to have a fragment at this G. Okay, you're going to have a fragment at this G. So that's the fragment that's going to be left. You're going to have another fragment, and then that fragment is left here. Okay, and remember, this fragmentation takes place on the 5' end. In the other case, which is the first reaction that we looked at, and that is the depurination, you're going to have a reaction with the G. Some of it is going to get cleaved. So you're going to have the G on the 5' end cleaved. You're going to have another G on the 5' end cleaved here. You're going to have an A also getting cleaved. So you're going to have a random set of A's and G's getting cleaved. So you're getting all these fragments. Now that reaction vessel consists of all these fragments. Here you have defragmentation, which means you're going to have the A's and the T's fragmented. So you're going to get various fragments of various sizes. And similarly for the C, okay, you're going to see on the 5' end. So now you're going to get these fragments, okay, and these fragments are going to chromatographically separated using electrophoresis. Okay. And now these are the fragments that you see. So you have a stationary phase and you have a mobile phase. And the stationary phase is what holds something back, and the mobile phase allows it to flow. So something that's large is going to be held back. So that is the fragment associated with the full sequence. And then you have one fragment which goes down a little and the other fragment which goes down a little more. Similarly here, what you're going to have, first of all, there will be some DNA which is not going to get fragmented. And because it's not going to get fragmented, you see across the board, this is going to be the full fragment. It's the largest sample, so the stationary phase is going to hold on to it a lot more, and it's not going to get washed out. So here you're going to have another fragment, another fragment, another fragment, and same thing, you have a lot of break here, depending upon the different kinds of fragments you have. And so what happens now is, when you look at these fragments, you could potentially have on this side, okay, you could potentially have on this side. So for example, you see what you have here is an A, okay, an A, and that's the smallest fragment. And then you have an A, and then you have an AT. But you know specifically where the breaks occur here, so you know that there is an A and it's attached to a T, so you know there's an AT fragment. Okay, similarly here, if you see this is the smallest fragment, ATTGA, then you know that that's the fragment takes place when you cut at the C. Okay, so you know that you can put an AT and then you put an ATTGA, but then here you have an ATT. So now you know that an A is attached to a T, and then because you know exactly that ATT is attached here, and that also tells you that an ATT is attached here. Okay, so that means you know now you start building the fragment, building your sequence from the fragments. And now this is ATTG, and so you know there's an A, an AT, an ATT, so that's one. So you know that's ATTG, and then you know that there is another A here. Okay, and so what happens is, once you have these fragments and you can determine the masses of these fragments, because you know exactly what size is associated with what that particular size of this fragment, what it is composed of, based on these mass differences on how far this particular fragment is held on the stationary phase. And then this is obviously the one which is the slowest, which is obviously the biggest one. And then you can reassemble. So think of it as a linear jigsaw puzzle that you're putting together. Now, what's this fifth point, that is the radioautography, is because you can then kind of, when you assess the sample, because your primary fragment here is, because of the primary fragment here is radioactively labeled, it gives you a radioactive signal. Okay, and so for example, if you develop this particular gel on a photo plate, The radioactivity is going to mark that particular plate, and that's when you're going to see these signals. And then because we know exactly what the masses of some of these fragments are based on fragment by fragment by fragment, you can then reassemble this as a kind of a linear jigsaw puzzle. So that's the Maxam-Gilbert. Now the Maxam-Gilbert fell out of favor because the Sanger sequencing came along, and Sanger sequencing turns out is a superior sequencing method. And the Sanger sequencing method was published a little earlier, and it is what is called the chain termination method. Now, the fundamental difference between the Maxam-Gilbert and the Sanger sequencing method, the fundamental difference between the two is that the Sanger sequencing method builds a protein sequence, okay, whereas the Maxam-Gilbert sequencing method fragmented and then kind of put all the fragments together in some sort of a kind of a systematic, intelligent jigsaw puzzle to determine what the sequence is. And we'll see what building the sequence means. So once again, the concept is different from that of Maxam-Gilbert. Rather than break up a DNA sequence using chemical methods, the formic acid for the depurination, the hydrazine for the depyrimidation, the dimethyl sulfate for the methylation, a DNA sequence is selectively built up using the enzyme DNA polymerase, starting from a DNA primer. Okay, now here's an example of what a primer is, and we can read this. A primer is a short nucleic acid sequence that provides a starting point for DNA synthesis. It allows, it's a short sequence, and what DNA polymerase, for example, begins the building of this DNA sequence by creating nucleotides that are complementary to the nucleotides on the primer. Okay, in living organisms, primers are short strands of RNA. A primer must be synthesized by an enzyme called primase, which is a type of RNA polymerase, before DNA replication can occur. The synthesis of a primer is necessary because the enzymes that synthesize DNA, which are called DNA polymerases, can only attach new DNA nucleotides to an existing strands of nucleotides by building the complementarity. The primer therefore serves to prime and lay a foundation for DNA synthesis. Now that's like priming the pump, right? So if you prime the pump so that it can draw fluid from, for example, a well or some sort of a receptacle, what you want to do is you want to be able to fill that tube with water, right? So the capillary action can actually help draw the water once the pump starts working, so it doesn't. For example, if you were using your some two-stroke engine thing like a weed whacker or something like that, and when you put your gas oil mixture in there, there's a little tube that you pump the tube a few times to fill that tube with the fluid. That means all the pipes are filled with the fluid, and that's why when you run whatever that machine is, it ensures the flow of oil as more or more fuel goes into that. It ensures that the lines are full. Okay, that's kind of the notion of a primer. The primers are removed before DNA replication is complete, and the gaps in the sequence are filled with DNA by DNA polymerases. In the lab, scientists can design and synthesize DNA primers with specific sequences that bind to sequences in a single-stranded DNA molecule. These DNA primers are commonly used to perform polymerase chain reaction to copy pieces of DNA or for DNA sequencing. Now, this is, it's not easy in the sense that to design the primer that somehow involves a little bit of a guesswork. It involves a lot of research. It involves, looking for a similar DNA sequence. If you're looking kind of to build a sequence related to a particular species, you're going to look for a related species. But this is a good working definition. Okay. What is DNA polymerase? Let's look at another definition. Again, this nature.com side table definition gives you a lot of, quick and dirty definitions. For example, if you're working on something and, there's a bunch of jargon in there and you don't know what that means and you want to have a good understanding, this is a great place. So let's say DNA polymerase is a type of enzyme that is responsible for forming new copies of DNAs in the form of nucleic acid molecules. Nucleic acids are polymers which are large molecules made up of smaller repeating units that are chemically connected to one another. DNA is composed of repeating units called nucleotides or nucleotide bases. DNA polymerase is responsible for the process of DNA replication, during which a double-stranded DNA molecule is copied into two identical DNA molecules. Scientists have taken advantage of the power of DNA polymerase molecules to copy DNA molecules in test tubes via polymerase chain reaction, also known as PCR. So, you are building your using the primer and using DNA polymerase, you are building your DNA, which is complementary to the sample that you're trying to sequence. Okay. But the question is, well, you still have to build fragments, because the idea of putting this linear jigsaw puzzle that we saw in the Maxam-Gilbert reaction is about the same as in the Sanger sequencing, except you're building fragments. But now, you want to still create those fragments, but rather than take your starting DNA sample and fragmenting it, you're building these DNA fragments. But at some point, you want that fragment to end so you can actually put it together. And that's why you're blocking the buildup of these fragments using what are called dideoxynucleotides, and we'll see what that is soon. So, much like the Maxam-Gilbert method, we end up with fragments rather than breaking an existing sample into fragments. And I have to repeat this just so we understand the difference between the two. The fundamental conceptual difference between the two methods is that you build these fragments, you terminate the creation of these fragments, and then you put the fragments together. And the fragments are built complementary to the DNA sequence that you're interested in. So we end up with fragments which can be subjected to gel electrophoresis and fluorescence-based methods to detect the bands. And the bands are associated with certain mass, certain fragment sizes. And then, again, these are the fragments that you put together in a kind of a linear jigsaw puzzle that I talked about. In this way, the fragments can be used to put the DNA sequence back together. And once you put that in the right order, what you see here is the beginning of the discussion of a dideoxynucleotide. So, for example, you have your ribose sugar, you have your phosphate group, and you have the purine or the pyrimidine. In this case, it's the purine. What we are really concerned about is with this hydroxyl group. So, to continue the discussion, what you see here is a deoxynucleotide triphosphate, which are then used to build the nucleotides. And when you deoxygenate this OH group and you have a hydrogen group, remember, this is where you had the OH group in ribonucleic acid and you replace it with a hydrogen. When you take away the other OH, the other O from the OH group and replace it with a hydrogen, then you have 2-deoxy or dideoxynucleotide triphosphate. And it is this molecule which terminates the chain in Sanger sequencing. The total sequencing step contains 4 sequencing vessels: the DNA template, whose sequence is to be determined, DNA polymerase, a primer, and the nucleotides. To each vessel is added the dideoxynucleotide triphosphate for adenine, guanine, thymine, and cytosine. And then the deoxy are added in excess to their di counterparts. So remember, we need to build these fragments. And when, for example, instead of a DATP, a DDATP gets added on, that fragment is completed. Okay, because of the dideoxy nature of it, that fragment, DNA polymerase cannot add another nucleotide to it. Okay, so you have more of these because you want the sequence to be built, but then you want it to be terminated. But you don't want the sequence to be the building of the sequence to be terminated very quickly, and that's why we want to make sure that this is not added in excess. Okay, this allows for fragments of different sizes to be formed before an interaction with a dideoxynucleotide fragment occurs to terminate the chain reaction. The resulting fragments are denatured, and then subject it to gel electrophoresis. So we'll go to this particular web page, and it's a short cartoon demonstration of the Sanger sequencing.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into two strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
The Sanger sequencing summary of the protocol. So you have, this is a single-stranded DNA. This is the one whose sequence you're interested in finding out. Then you have a primer, and remember the primer is a short sequence which is complementary to the beginning of your sequence. Then you add DNA polymerase, which catalyzes the formation. You have an excess of the deoxynucleotide triphosphate. And then to that you have, for example, so all of this will contain, you'll have 4 vessels containing all of this. And then you will have, in each vessel, you're going to add the dideoxynucleotide triphosphate. And so what happens is, as this begins to build up, the moment you have an A, because this is the dideoxy, it prevents further reaction. And so what happens is, wherever there's an A, so when you look at these fragments, you'll realize that the termination occurs wherever there's an A. So you know exactly what the rest of the fragment is because you know exactly how it ends. Okay, so for example, wherever you have a DDTTP, your chains will terminate in a T. Okay, and so these are the different fragments that you're building up. And now because you know exactly what the masses of these fragments are and how each of these masses are different, you know exactly what's added to what. So you have the sequence of interest. You use this to build a complementary sequence, and using the same electrophoresis method, you will be able to build the complementary sequence. And from the complementary sequence, you will put the complementary sequence together. Now it's not error free, but it's got pretty good error efficacies. Okay, and you're going to be able to build the complementary sequence this way.